This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 43, recorded on October 3rd, 2012. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hi there. How are you?
I'm well. How are you doing?
I'm doing fine. I'm busy.
I think you're always busy, right?
I know. It's. I think I told you once. My wife doesn't know if I'm retired or retarded.
Yeah, you're fine. I think it's good to stay busy. You're not really retired. You just change jobs.
I guess I'm not getting paid as much, but that's okay.
Yeah, well, retiring.
I enjoy what I'm doing.
Also joining us today from the Medical University of South Carolina, Michael Schmidt.
Hello, everyone.
How are you?
I'm doing well. I'm looking forward to today's topic. It should be fun.
Always good stuff, right?
Always.
How's the weather down there in Charleston?
Well, I think it's thinking about raining. It hasn't quite made up its mind. It's very swamp-like. This is definitely not Chamber of Commerce weather for October. It's hot and muggy, and they're talking about a cold front, but unlike the upper Midwest where they're predicting snow for tomorrow, they are just predicting that we'll be in the mid-70s with sunny for the weekend.
Well, here in the Northeast, we're in the rain mode. Started yesterday, raining all today, tomorrow, Saturday, Sunday, 22 degrees Celsius. We need the water.
Yeah, we definitely need it here in South Carolina as well.
Today we have two papers. We're back to the paper mode here on TWiM, but these are two very cool papers that you'll want to listen about and maybe even go and read. And the first one, Elio found and is very excited about. It's a paper published in Cell, and it's called Constitutive Formation of Caveolae in a Bacterium. And caveolae is a kind of Italian pasta, right?
Yeah, right.
My grandmother...
It reminds me of the one that looks like the ear.
Orecchia.
Yeah, I can never pronounce it.
Right, Elio?
Which one? I don't know.
Little ears. Actually, it's
orecchiette.
Yeah.
Orecchiette.
Caveolae are the ones my grandmother used to make. We're just kidding.
I'm getting hungry. Anyhow, so you want to hear about caveolae in bacteria, don't you?
Yeah, I'd love to.
This is an amazing paper. I mean, we often hype up with the papers we discuss, and we like to say they're all terrific, but this one is off scale. Here's the story. People have cloned a single protein from animal cells into E. coli, and that E. coli now makes intracytoplasmic vesicles, which are wall-to-wall vesicles. In other words, the bacterium is full of these endocytic vesicles. Now, back off a minute. You may remember, or maybe not, that the original definition of prokaryote by Van Niel and Roger Stanier included prokaryotes do not carry out endocytosis. Mm-hmm. So there goes this one. Anyhow, let me tell you what the story is. So the protein in question, and it's a single protein, okay, that's it, cloned into E. coli, is called caveolin-1. Caveolin-1 is the main structural protein of things called caveolae. Caveolae are indentations in the membranes of eukaryotic cells where a lot of business is transacted. A lot of things happen there, okay, including signal transduction and all kinds of processes, including also the business of bacteria and viruses entering into cells. So caveolae are hot stuff in eukaryotic cell biology. Now, so if you look at them, they look like little flasks protruding inside from the cell membrane into the cytoplasm. Okay, they're little urns. They're about as small as you can make a membrane-bound vesicle. There's only so much curvature you can put into it. And anyhow, so now they cloned this one protein into an expression plasmid in front of a high expression promoter. That's the promoter for T7 RNA polymerase. So lots of protein is made. Lots of protein. Man, the cell gets filled up. And that's it. It's the only protein that's made. Now, in addition to making the protein, they got to make lipids. Okay, so there's a huge amount of lipid synthesis. Now, like my buddy Mary said, it sounds like the Sorcerer's Apprentice. I mean, this gene gets going, and man, does it get going. I've never seen anything like it. There's nothing like it that I know of in all of microbiology. Seeing a cell which is loaded with these vesicles. Okay, that's the general setting. Do you agree this is phenomenal?
I think it's off the hook phenomenal, because if you think about the translational applications, if you could now begin to incorporate drugs into these little tiny vesicles, you could have the ultimate delivery vehicle.
We're going to get to that. That's exactly the point. Yeah.
Let me add one thing you mentioned. So this is a eukaryotic protein.
A human protein.
Human protein. And these caveolae are part of what's called caveolae-dependent endocytosis in eukaryotic cells. And
that's right.
They bring things in from the extracellular. But viruses also tap into this. So there are a number of viruses that get into cells by caveolae-dependent endocytosis, as opposed to clathrin-dependent or other kinds of vesicular endocytic pathways. So people study these a lot in eukaryotic cells, figure out how they work, and maybe use them or interfere with viruses getting into cells.
Yeah, they're a hot subject. There's a lot of work is being done, but I have no idea what got them to think of putting this in bacteria, because anybody who they would have asked, any microbiologist would said, good luck, but it ain't going to work. So
it's probably one of those things you're sitting around a table and someone says, why don't you try expressing in bacteria? Just take you a few days,
Right.
And it works.
Anyhow, so this is, so now these structures look and smell like the caveolae of eukaryotic cells. They have only one protein on their in the membrane, that is caveolin 1. They lack regular cytoplasmic membrane proteins. In other words, this is a very highly selective mechanism. The lipids are a little bit different, not hugely different, but a little bit different than the lipids of the inner membrane, possibly having to do with the curvature, because these guys are little spheres, and the curvature of the membrane is about as extreme as you can make. In other words, you can't make them any smaller. I think maybe you can, but not easily normally. So the reason they did this is because these people are interested in the structure of caveolins, and they thought appropriately that working on E. coli would be simpler. It turned out to be simpler. It turned out to be simpler in spades. This is the E. coli turns into a machine for making caveolae, period. So they can isolate them by breaking up the bacteria. They simply sonicate them, treat them with lysozyme, etc. The caveolae are, of course, pretty tough because they're small, and that adds for a great deal of stability. So they can isolate them, they can purify them, they can do a lot of things. Before we get into the point that Michael brought up, this is a paper that stands out as being a great surprise. And by the way, also they catch caveolae in the act of being formed off the cytoplasmic membrane. And it looks exactly like what happens in eukaryotic cells. In other words, they look like buds, inward buds.
The cool thing that's different here, of course, is that when it buds in, it's not open to the extracellular space.
That's right.
Because there's another membrane on top, right?
Right.
So it's unusual because it just shows you just need a membrane anywhere and it will butt in. So even if there's something covering it, it's...
Sure. But what is it about caveolin or the gene for caveolin that just gets going and going and going? So anyhow, so the paper, of course, has, as we'll see in a moment, has tremendous potential for other reasons, and it's interesting for caveolologists to study because this is a nice system. But from a microbiological point of view, it raises a huge number of questions. And I got to tell you that the paper was very, very, very thin in what happened to the bacteria. So I wrote to the lead author. Sorry, I've forgotten his name for a second. It'll come to me.
Piers Walzer, or Robert Parton?
Walzer. He was very gracious. He acknowledged that they should have paid more attention to the bacteria, but there's eukaryotic cell biologists after all. Anyhow, he assured me that the only gene cloned into this was the gene for caveolin. It was the pure, unadulterated, clean cloning, except when they wanted to add GFP or make fusion proteins with the caveolin, but that's it. There was no other regulatory element, nothing that they knew about. And what's more, so I said, I wanted to know, do the cells die when they make this? Well, he didn't really say, but he did tell me that when you inoculate them in broth, an overnight culture of the cloned construct is as thick, has as many cells as the wild type. In other words, they grow like crazy. Okay, and where they probably even make colonies, but I don't know where there's no room for anything else. I mean, I don't know where the ribosomes are. I don't know where the DNA is. It's just really, when I say, totally displacement of the cytoplasmic content. So to a microbiologist, this is really, it's an artificial system, granted. We may not see this in nature. Nobody has yet, with one exception. There's a claim that planktomycetes, which are sort of a far-off group of bacteria, may carry out endocytosis, but the details are not known yet, and it's nothing on this scale. So artificial or not, it talks about the plasticity of a bacterial cell. That's the least it does.
Do these cells get larger as a consequence of all the vesicles in the cytoplasm?
I don't know.
Their EMs didn't look like they were making giant cells or elongated cells. I mean, the EMs in the paper, it just looks like garden variety E. coli chock-a-block full of vesicles.
Yeah.
The overnight ones, the ones that are totally full, may just be bigger, but just how much bigger, I don't know. So tune in, because there's going to be a lot of microbiology to do here. As I say, the fact that it's an artificial system is in a way an issue, but so what? I mean, some artifacts are wonderful, right?
Well, these were grown in E. coli BL21, lambda DE3 lysogen, and the plasmid was driven by a T7 RNA polymerase promoter, and I'm trying to remember the control circuit of BL21 lambda DE3 to know if that promoter for the T7 RNA polymerase, which is in E. coli BL21, is constitutively expressed so that it's always on, or whether it's induced by IPTG.
Well, they induced it, but then they kept it induced.
During the overnight event.
They just kept it induced. And they didn't do any studies about pulse induction or something like that. What happens when you remove the inducer? No, this is just not, that kind of microbiology hasn't been done yet, apparently. Although Dr. Walzer assured me that they're interested and that they will pay attention, and hopefully in future papers there'll be more details of the microbiology done.
So one thing I found interesting, Elio, is so there's another caveolin in eukaryotic cells, number 2, and that doesn't work in E. coli to make the vesicles.
No, it makes strange little structures, but they really don't look like caveolins.
Neither does the C. elegans version of the protein. It's very interesting.
It's funny, isn't it?
I mean, you'd think if it worked in bacteria, it would work in worms, right?
Yeah. Well,
I mean, the worm version would work as well.
It probably is a function of the cytoplasm and maybe the overall, as Elio said, where are the ribosomes? Where's the other stuff inside the cell? And while we may think of bacterial cytoplasm as being a reaction vessel that's principally water, it's really chock-a-block full of proteins and lipids and doing the business of making more bacteria. Plus, let's not forget the RNA and DNA.
Mm-hmm.
And when you cram all of this stuff into the small space, the octanol water coefficient, when you begin to make these lipids and you make this massive amount of protein under T7, may cause one version to fold aberrantly over the caveolin. So again, this paper is an illustration in scientists must be lucky.
You're not kidding. This is amazing. So anyhow, it's the paper goes on and asks whether you can introduce proteins into these caveolins, these artificial caveolins. So the answer is yes. Otherwise, I wouldn't tell you. And this is what they did. They started out by, first of all, they put some dyes in it, and fluorescent dyes that cross the membrane, of course, end up staining the caveolae and all that. All that's very nice. And also hydrophilic dyes end up within the caveolae. But they added horseradish peroxidase, an enzyme that doesn't belong to E. coli, and got it into the periplasm by loosening the outer membrane. And not many details about that, but anyhow, they got it into the periplasm. Well, if a protein is going to be in the periplasm, it's going to be part of the contents of the caveolae. And sure enough, that's what they found. Because as the caveolae bulge inwards, they're going to be taking up protoplasmic material. And they did just that. So they, and it's active. So they take the caveolae, isolate them from this manipulated strain, and inside they find horseradish peroxidase. So it works. It's a delivery system to the inside.
Are there other molecules in the periplasm that you would expect to be into the caveolae?
You would, and they don't say. I try to look and see if they find alkaline phosphatase and sugar-binding proteins and all the good stuff that's in the periplasm. However, it may be that there's only so much of that in the periplasm, and as you make more and more and more caveolin, you dilute it out, so that eventually the last caveolins to be made end up just with buffer of the periplasm, with the small molecular weight solutes, because the proteins have been depleted. But be that as it may, what they did then is to make a fusion, to say, is this a delivery system for, let's say, cancer drugs? The argument, I guess, is that caveolins would be better at delivering proteins than liposomes because they are more natural. I don't know. Actually, I don't know anything about the delivery of drugs by little vesicles, artificial or otherwise, but I can imagine that it works better with caveolins than it would with just lipid membranes or any other construct. After all, this is a human protein, so it even sounds good when it comes to down the line attempts at introducing a drug. Anyhow, so they fuse to the caveolin. By the way, the caveolin is very forgiving. You can fuse all sorts GFP and other things to it, and it still is active. So you fuse a domain for an immunoglobulin to the caveolin, and now you purify the caveolins and you add them to essentially a cancer cell which has receptors for that domain. And guess what? The protein gets in. The domain, the immunoglobulin domain fusion gets in. Okay? So talk about luck. I mean, that didn't have to work that way. It seems to work quite well. Early stage studies, but very, very, very much in keeping with what Michael just said earlier, that this may be a phenomenal delivery system for drugs because it is in such an unusual vesicle that lends itself to such studies.
So in that case, Elio, where they delivered material, are the caveolae fusing with the plasma membrane?
Yeah, I think so. Let's see, I don't remember what all they did about that, but there is not a lot of detail yet, but I think that, I think that that's what they expect to happen. Certainly that's what I would expect to happen. Otherwise, how would they get in?
Well, they comment the findings that they present, and this is their quote, presented here provide the first characterization of a potential drug delivery vehicle that would represent a low-cost submicron drug encapsulation method that can be engineered towards specific cellular targets. And so they call it a genetically encoded vesicle. And I think, if you think about how to make drugs and all of the controversy that's surrounding nanoparticles and nano delivery, this is for better or worse, a human protein that is being used with a fusion molecule to target a specific event. And you can put it in, and it will probably have low toxicity because of the targeting specificity with your fusion protein, specifically going after the cancer. So you don't have all the associated side effects that we see with many of the cancer drugs that are just killing indiscriminately. So I think, this is probably going to be recognized in a few years as a seminal paper in showing how to understand fundamental cell biology at a eukaryotic level, and then looking at a way of taking advantage of it in a prokaryotic sense to understand how you can make a drug that you can then resend back for patient use.
That's right.
So what happens here at Elio is that the caveolae are internalized themselves, and then they end up fusing with other vesicles in the cell, which they would normally do.
It's not known, but this sounds like a plausible scenario.
So they say binding internalization and progression into multivesicular bodies was observed.
They haven't done. They don't show much data on that, but that's probably what happens. I mean, it's more than likely, and so it's perfect. It's absolutely perfect, and I would buy stock in the caveolae company.
Yeah. So Vincent, let me ask you about our friends, the eukaryotic viruses that use this particular system to get into the cells. Has that biology taught us anything remarkable in terms of specific targeting, or is it really the expression of the viral program that then targets the caveolin vesicles too?
Yeah, well, so the ones that are best studied, as far as I know, are the polyoma viruses like SV40. These tend to get in by caveolin-dependent endocytosis, and they don't seem to modify the program. They just use it. And the curious thing about at least some of the caveolae is that they target the endoplasmic reticulum.
Oh.
So in clathrin-dependent endocytosis, eventually the vesicles move into the cell and they fuse with lysosomes. And many viruses get out before that step. But these caveolae seem to target the ER, and that's different. And so people are trying to puzzle out how that works, and in particular, then how you would get the viral nucleic acid from the ER into the nucleus.
That is true when Legionella gets into cells. The vesicles surround themselves with endoplasmic reticulum. Well, I would have guessed that the idea is to pour in more protein needed.
Yeah, like a vacuum.
The bacteria. And the bacteria, it makes sense.
Yeah.
Viruses, I don't know.
What's neat about this is that in eukaryotic cells, it's thought that caveolae need cholesterol to be stable. And of course, there's none in
There's
coli.
none in our friend the bacteria.
So they have clearly their membrane is different enough to support the formation of stable caveolae.
Yeah. By the way, the paper has one interesting aspect of that. They did the lipidome of the caveolae and compared it with that of the cell membranes. The lipidome, it's really using mass spec and things to really get into depth about the kind of lipids you have. So they don't just look at the difference in polar head groups, ethanolamine, phosphatidylethanolamine, phosphatidylglycerol, but they also look at the length of the fatty acids. So this gives them a very large amount of information. Out of it came that, yes, the caveolae have slightly different fatty acids, like they have lysophospholipids, for instance, but not a lot jumps out. Like they thought, actually, in correspondence, he told me that they would thought it would be more cardiolipin, because cardiolipin is a curvigenic lipid. It induces curvatures, but it didn't really. So they need to do more work about it. But they are very savvy. I mean, this is a group. It's 16 authors from 3 continents. The main ones are people working in the University of Queensland in Brisbane, Australia. But it's a large group, and so they have the expertise it takes to do image analysis towards protein structure, lipidomics, and so forth. So, that's what they needed is more microbiologists, I thought.
Yeah. Well,
maybe some of these people are. I don't know who all they are, but it's possible. But their presence was not felt in the writing of the paper, let's say.
One of the last things I'd like to say is in their supplemental information, they have some movies in lieu of figures. And it's the first movie S1, shows the constitutive formation of the caveolae in the host cell. And I think this is one of the distinct advantage of electronic publications over print, because the movie actually takes you through the focal plane, watching the tomographic reconstruction showing the expression of these caveolae in E. coli. It really drives home how this poor cell is chock-a-block full of these tiny little vesicles. It's really neat to see.
I got excited. I got to admit, I'm usually very cool when it comes to science. I got very excited.
By the way, did you know that caveolae were discovered in 1953?
My God, that far back?
It's the year I was born.
Also, as old as you are.
Also the year that DNA structure was solved, right?
Yep.
Announced.
Announced. So what do you mean? It wasn't solved that year?
Well, no, it was solved before.
Yes, announced. Okay, yeah.
Yeah, it was announced in the States. Yeah, in fact, I was there at Cold Spring Harbor when Jim Watson got up and showed how this was a double helix.
Oh, yeah?
Yeah.
What did you think at the time?
To my eternal shame, I didn't see what the big deal was. So the double helix is just another shape. Took me a while to digest why this was important. Yeah, because if you think about it, so it's a shape of some sort.
Yeah.
So, but I got excited when I figured out that this explained how DNA replicates.
Yeah.
Then it got my attention. But what did I know? I was a graduate student.
Yeah, I remember when someone told me about RNA splicing, and I didn't know what. So what? What's the big deal? But I was also a graduate student, so it took a while to sink in. I still don't quite understand. But
the first time I heard of splicing, it gave me a headache. I mean, I said, thank goodness I'm a bacteriologist, so I don't have to worry too much about splicing. But then I learned bacteria can also do splicing, so then I had to go and figure it out. And it's really the whole issue of splicing by itself is an interesting topic.
Very. Okay, that's a great paper, and maybe it will push our understanding of caveolae forward. Should. It's great.
It has already.
Yeah, a couple of people have pointed out the paper saying this is amazing. So it's one of those things. All right.
It's on our blog, by the way. It's in our current version of Small Things Considered.
We will link to it for sure.
Okay, thank you.
Our second paper was pointed out to me this summer. I ran into Jeff Miller, who is a microbiologist out at UCLA, and he happens to be the current president of the American Society for Microbiology. And he was saying, we have this paper in press where we isolated phages of the acne bacteria. And so now it's come out in MBio. It's called Propionibacterium acnes. Bacteriophages display limited genetic diversity and broad killing activity against bacterial skin isolates. And so he said there were dermatologists involved in this, which of course you would expect for acne. And besides Jeff Miller, there is Graham Hatfull, who is a phage guy in Pittsburgh. And I guess he was involved in the sequencing and a bunch of other authors as well. And
Some
acne,
of the comparison to other phages also.
yeah. So of course, acne is a big deal. If you search the Google for acne, you get hundreds and hundreds and hundreds of therapies and websites telling you what to do, because as kids move into puberty, they many of them develop acne, and it messes up their lives, makes them insecure and unhappy. And so there's a lot of impetus to try and do something.
Plus, it physically hurts.
It hurts.
You got to remember that what's actually going on is you're actually forming granulomas,
Mm-hmm.
and
Right.
you're recruiting all those damn pus cells. All the white blood cells are coming in. There's not one of us that has not popped a zit.
That's great.
And you can actually feel the granuloma as you begin to move that pimple around. And to give our listeners who are not microbiologists an idea that you've actually experienced Propriano bacterium up close and personal, is if you actually, as you're moving your fingers away from your nose or your face where the pimple was, you can actually get a whiff of what smells like Swiss cheese, and it's actually the propionic acid that this microbe makes as its ultimate electron acceptor. And hence its name, Propriano bacterium. They actually make propionic acid.
It's
And
great advice. Go smell a zit.
go smell a zit, and you'll actually discover this remarkable microbe up close and personal. And there's a variation of Propriano bacterium, a granulosum, that is also, and that's typically responsible for the cratering form of acne, where you get the deep divots in your skin.
Right. That's another problem. It can scar, and
Yes.
you got it for the rest of your life. So this is this bacterium is normal skin flora, right? And
Yes.
And then at some point in your life, it begins to, I guess, the numbers go up, and you get inflammation, and that leads to the acne. Although not clear to me that it's completely understood, right?
No, it's really a complex dynamic. A lot of it has to do with the complex immunology associated with the skin. And since we're in flu season, everybody has probably seen the commercial for the new flu vaccine that actually uses the dendritic arm of your immune system, with the short little tiny needle to elicit the antibody response, to inoculate yourself. And if you think about it, the life history of Propriano bacterium is it grows in this hair follicle, the sebaceous gland. You're feeding it oils. And what happens during puberty, as we all recall, is our skin gets much more oily. And so what we're doing is we're feeding the zoo animals, and they're making more zoo animals. And the net consequence is immune surveillance may go up, and we end up developing the inflammatory response that can result in the formation of these common granulomas that manifest themselves as abscesses or pimples, if you will.
So this paper, I think, one of the ideas was to look at the microbiome of this hair follicle, essentially, because the P. acnes is the main bacterium there. It's called the pilosebaceous unit. It has the follicle and the muscle, the hair, the glands that produce the oils. And so this is the main bacterium, and they wanted to see if there's a main species in this very, very specialized microenvironment. Is there limited diversity in the phages? That's one question. And the other is, could you use phages to treat acne, because the existing therapies are somewhat problematic, and if you use antibiotics, then of course you get resistance as well. So they went about isolating phages of P. acnes, and there have been some isolated before, but they went after many more. I don't remember the number now, 9 or 10, 11 phages they isolated. They isolated these from bacteria recovered from individuals, from their skins, from people with or without acne. And they also recovered phages from a culture of the bacterium that's stored at the American Type Culture Collection.
That in itself is kind of interesting
Mm-hmm.
because you don't necessarily expect that an old culture would carry phages.
Mm-hmm.
You know, and, well, how come? You know,
Yeah.
it's not obvious to me that this had to happen, but it did, and it's a good thing.
It's probably because it was a clinical isolate, I would bet, and they probably pulled it out of this specialized niche. And what this paper reveals to us is that these phage are ubiquitous in that microenvironment, and when they pulled the microbe out and amplified it, they similarly amplified enough of the phage to get deposited in the culture collection.
Yeah.
Well, still, I don't get it, but that's okay.
So they would grow up these cultures and then take the supernatant and then inoculate them onto P. acnes monolayers, and they would get plaques. And so many of them were lytic phages, that is, they made clear plaques, and they say a few of them were turbid plaques as well. So they have very nice electron micrographs of these phages. They all look very similar, and they have this so-called cyphoviral morphology. There's a head, a icosahedral head, and then there's a very longish tail at the end of it. And they all look very much the same. So all right away, just looking at them, they're all quite similar. And then they sequenced all of their genomes. So they sequenced 14 different phages. And the genomes are all pretty much the same size, around 29,000 base pairs in length, 45 to 47 protein coding genes. And when they begin to analyze the DNA sequences, they find that, in fact, they're really very similar. So again, these are 14 phages isolated from different individuals, geographically separated, temporally separated, 30 years. One of them was obtained 30 years ago. They're all very similar in sequence. The GC content is very similar among them. The gene organization is very similar. So there's very limited diversity in all of these phages, which they find surprising. And they think maybe, it's related in part to the fact that this is a very specialized microenvironment, and therefore the phage populations are similar.
Well, I want to ask you this question. You may get to it in a little bit, yeah. Vincent. Do you think the lack of genetic diversity in this phage population is specifically targeted, coupled with the fact that these phage don't form lysogens, which is typically seen in a large number of phages in the bacterial that infect bacteria? Do you think the phage are actually keeping the P. acne population under control to effectively keep it under the surveillance radar of the human immune system, so that you actually have just the right number of P. acne to control the level of oil that the sebaceous gland is dumping out to prevent other microbes like the staphylococci and the streptococci, which when they become locally invasive can become a life-ending infection.
Right.
Whereas P. acne typically does not become invasive, nor does it become a life-ending infection. So the phage are actually enabling this complex life history of P. acne to carry on.
Yeah, I think that's a really interesting possibility, which they have no data to address at all because they didn't really. I mean, you'd have to look in the gland and look at the production of phage and see if it in fact is made during a certain time and it's limiting the bacteria. But you could do that. It's an interesting idea. Sure, why not? In the oceans, phages limit populations of microbes as well.
Well, we also have the best example would Vibrio cholera, where phage actually controls cholera outbreaks. Just by the mere concentration of phage that's in the monsoon rainwater determines whether or not you're going to have a cholera outbreak. So,
yeah. One of the interesting things they do find is that many of the proteins are different from those of known P. acnes phages. So brand new, every time you sequence new phages, you get new genes. And it's the same thing here, and you don't know what they do. They do find phage proteins that seem to be involved in cell lysis of the P. acnes, lysins and holins and so forth. So those are present there, and those are recognizable. And the other thing that's quite interesting is that then they take all of these phages and then they ask if we infect different P. acnes hosts with them, what happens? So they're looking for phage sensitivities. Are the bacterial hosts sensitive to all the phages or not? One phage, which they recovered from an ATCC strain, infected all of the P. acnes hosts. Okay.
And that's a very profound statement. Having worked in phage biology for about 6 years, that was one of the conundrums we faced when we were developing our thinking on making ethical pharmaceuticals based on phage, because so many bacteria became resistant to phage. Right. So you had to dope your cocktail and literally have a cocktail. By virtue of the fact that one phage can actually infect many different species, you get around the population bias that is really limiting the utility of phage as an ethical pharmaceutical.
Mm-hmm. Interesting. Yeah. So they find that two P. acnes strains are resistant to 9 of 10 of the phages. All right. And they find that this resistance correlates with the presence of specific CRISPR loci. We haven't really talked about CRISPR here on TWiM, have we? Although we've talked about talking about it.
We had, I think we did one episode very early on.
Yeah. CRISPR clustered, regularly interspaced, short palindromic repeat elements. And these are sequences in many but not all bacteria that are believed to be a sort of primitive form of immunity and which provide resistance to phages and plasmid DNAs that would be introduced and would be detrimental. And they seem to be sort of a memory, from how I understand, when a host is infected with a phage or picks up a piece of DNA, a bit of that DNA is incorporated into the CRISPR loci and then serves as a sort of template for degrading the DNAs later on.
Well, if you think about the whole business when you were first introduced into lysogeny, and you were taught on how the bacterium looks for an insertion site, and there's specific phage insertion sites on the chromosome. E. coli's most famous one is, of course, phage lambda, and all of the elegant work that Mark Potashny did on phage lambda. And we all then recall specialized transduction, in which you have an abortive excision in which you pull some of the host material out, but you can begin to see how what gets a mistaken cut can also leave phage material behind. And so if you think about these CRISPR regions, these short palindromic repeats, and think about the old Campbell insertion model of DNA, where it's effectively looking for the direct repeats and then the inverted repeats, and you get the what I always envisioned in my mind is reciprocal insertion. You can begin to see how this could be a form of memory that these things are left behind by accident as a consequence of a bad lysogenic excision. And
it's clear that you're right, and it's clear that we have to spend time on CRISPRs because it is, in fact, a fascinating subject. So I hope we do that, and don't just talk about it, but actually do it.
Yeah, we will.
Would be a great thing.
We'll get a CRISPRologist on.
Right.
A CRISPRologist. How about that? So basically, we have 14 P. acnes phages. The diversity is very limited, maybe because of this unique microenvironment in the skin. Some of them can infect all of the hosts. And so they suggest maybe you can use these phages to treat acne, or maybe use one of the proteins, one of the lysine proteins from the phages to do that. And that's where Michael comes in, because this is the man with real-world experience in phage therapy. So what do you think about this idea, Michael?
Well, they got lucky. Jeff Miller is a lucky guy, and he really did. He and his group, this long list of august authors, really are fortunate, because the endolysine works on, it's 95% conserved in all phage types. Underscore the word all phage types of P. acne. So if you're trying to design a drug, so endolysine is a protein, and if you can put it back in our T7 system, and if we want to use the put it into some grease where we can actually deposit it into this very specialized niche, the endolysine could actually be a very good candidate to put into a topical ointment that you could apply to the pimple to actually go after the bacteria before they get to a point where you get a pimple or a granuloma formation, to effectively just control the concentration of microbes in that area. But the trick is going to be when to use it, because
I was just going to ask you, when would you use it? Yeah.
Well, that's the trick, because inflammation can be a very fast response. You know,
you wake up and you have pimples, right?
Well, what's that? The movie My Big Fat Greek Wedding or something, where the bride wakes up on the day of her wedding with a big pimple, and her father says, "Put Windex on it," and he uses Windex for everything, and so consequently you have to understand when the endolysine would be applicable, or do you just use a small concentration, or you then have to consider, like all antibiotics, whether or not the P. acne population will then develop resistance to the endolysine, though I think the endolysine just dissolves the cell wall.
It may be difficult because, as I understand it, the P. acnes lives at the very bottom of the follicle.
Yeah.
It's a very protected site. And it may be very difficult to introduce anything into the bottom of the follicle, but it may work. I mean, who knows? It's something you gotta try, ahead of time.
Now, you can imagine, Michael, as a child moves into puberty and they start to develop acne, then you start using this on a daily basis and may not get rid of the inflammation, but then it may quell future outbreaks. Who knows? But they have to clearly do clinical trials. So,
well, that goes back to my original. That's what got me thinking about the thoughts on the niche in this specialized area. Is if you begin to hypersecrete the food source for these bacteria, which we know is the oil. This is why every mother tells their sons and daughters not to eat chocolate when they're in their teenage years, for fear the oils will, the oil from the chocolate will lead to more oil production in the teenager. And so consequently, just controlling the food source can control the number of microbes, but some people that just plain doesn't work. Good old soap and water, where they tell you to wash your face more often to dilute the concentration of oil, may also not work. But I think Elio hit the nail on the head when he told us that the niche of this microbe is deep very, very deep where the oil is being secreted. And so consequently, all the scrubbing and abrading is not really helping all that much unless you can get really down deep. And so I think the phage, if Dr. Miller and his colleagues can figure out a delivery system where you just may not have enough phage. And one of the first clinical trials I would do is take a population that is presenting to the dermatology clinic and ask the question, what is the concentration of phage in the general public, and is there variability? Because I think that will then tell us whether or not phage therapy could be a viable opportunity for control. Because if I don't have any acne and my phage titer is 100 times greater than my Propriano bacterium titer, then that's obviously telling me that I'm keeping it in check. And recall that for these this particular phage type, Vincent, correct me if I'm wrong, but I'm remembering that the burst size is between 100 to 300 phage, depending upon the microbe. Does that sound about right?
That sounds about right.
I use the rule of thumb. Burst size for one phage in a bacterium is about 2 orders of magnitude. And so that may then give us an idea. And if you find a population that has frank acne, that the concentration is lower, and you can do the studies with youngsters that have not entered puberty, measure the phage titer, and then just move in a stepwise manner, asking what the concentration is as the oils go up and correlate against oil. I think you may have an understanding, because this is such a remarkable niche, in that this microbe is the representative microbe in this particular microenvironment, which then led me to think, what about our old story on Helicobacter in asthma? Could phage then help with that whole problem, especially in youngsters? Could we bring, are we having too many phage that are clearing out our Helicobacter? And do we need to think about controlling, because that's a very specialized niche in our stomach?
Yeah. Well, I thought the issue there was antibiotic eliminating the Helicobacter, right?
Well, yeah, but there's phage for everything.
Yeah, of course.
There's even phage for mycoplasma, which is an obligate intracellular parasite.
Wait. Don't say that. It's not an obligate intracellular parasite in my book. It grows perfectly well on agar plates.
That's
true. Don't call it obligate, because it's an issue with me. I picked a fight with some people about it. So in my book, anything that grows on agar cannot be considered a strict obligate intracellular parasite.
That's true.
It's normally it is found in the body of hosts, but it's not. The word obligate is. Okay, sorry about that.
Okay,
it's one of my hobby horses.
A phage for all seasons.
Hey, all right.
So listen to this. So you're talking about how the bacteria are way down in the hair follicle. You know how they got the bacteria for this study? So that's the problem. You have to sample the bacteria from deep in the follicle. Well, they use what are called BioRay deep cleansing pore strips. So this is a product you can buy, which is supposed to get deep down into the hair follicle, and they use that to pull out the bacteria for this project.
They also use comedones. They use the squeezate, the stuff that when you squeeze, they used a lot of that, so that was easier.
So you can deliver maybe phage or endolysins there. But again, you have to try it, and I think this is quite interesting. So, Michael, do you think we should expect to see some trialing of this in the future at some point?
The market of one of the issues that we had when we were developing our phage therapy is the whole issue of cost. You have to be able to make this very inexpensively, and mass producing phage is always a challenge because of the regulatory hurdles that the FDA places in front of you. And then the other way the trials have to be done, at least in the United States, is you typically do an inferiority trial where they're going to compare, will this medication work better than, say, tetracycline, which is the current modality for treating people who have really severe acne.
Yeah.
And so consequently, it's really an issue of the economics of how to get this done. But
That's interesting.
well, what the FDA was going to require us to do is to sequence every lot of phage. And this was done, we were developing this about 11, 12 years ago.
What was that for?
We were developing a phage therapeutic, and we were specifically doing proof of concept to see whether or not we could do it. But our target was acne.
Mm-hmm. Oh, really?
Yeah, our target was acne. So I found, in full disclosure, I found this paper absolutely fascinating. And I remember having a discussion with Dr. Miller. He came and gave a seminar here about 10 years ago. And we were still working on it. And I think I was about to make a trip to a French cosmetics company, actually trying to sell our technology to them. To make the long story short, we developed the technology for a German pharmaceutical company that has been since bought by Novartis. So most of my intellectual property is sitting on the shelves of Novartis.
So you had a phage at the time? You had isolated an P. acne phage?
Let's see. I have non-disclosure issues.
Okay. Well, I guess if you're working on phage therapy, you would have had to have a phage.
Yeah, right.
So,
Right, Elio?
Wait. Before we leave the subject, I'd like to point to what Dr. Hatfield has been up to in Pittsburgh and elsewhere.
Yep.
Changing the subject a little bit. I don't know if you know about it. He has started a countrywide project involving college freshmen to isolate mycobacterial phages.
Right.
You know about that?
Yeah, we interviewed him on my other podcast.
Oh, you did? Okay, well, so tell us about it.
They get it from the soil, so they have students have their own project where they go in their backyard, they get some soil, they isolate their own phage. They can name it after themselves if they want. They have the genome sequenced, and then they annotate the sequence. So everybody has their own personal phage.
It's amazing. It's an amazing story because these are it's a total immersion project. I mean, you take college freshmen who may not even be declared any major, leave alone biology, and you put them to work, and they have to get into great depth. Yeah. It's a educational system that I think is extraordinarily powerful.
Yes, so it was episode 87 of TWiM, and the program is called the Phage Hunters Integrating Research and Education, PHIRE, P-H-I-R-E program. And so we called the episode A Fireside Chat with Professor Graham Hatfield.
Well, it turns out that this is relevant to the paper here because the information, I mean, they compare the variety, the diversity of P. acnes phages to that of mycobacteria, and they have all this huge database. So there's something to compare to.
Yes.
You know, and that's sort of, it shows you the value, even not just for pedagogy, but also for science of doing this work.
Yeah, it's very neat.
Well, one of the things I'd like to bring up is we have the salmonella outbreak in our recent news associated with nut butters, principally produced out of that one factory in New Mexico. And one of the companies that's out there in the phage therapy space has a FDA licensed product to actually control the spread of Listeria and E. coli 0157 in prepared foods by actually spraying, in the case of Listeria, they spray 6 phages on food prior to preparation. And many of you know that Listeria is a big issue associated with prepared deli meats like turkey and other things, leading to large recalls. And so this company, Intralytics, which I'm not a part of, so there's no conflict here, has the first FDA licensed phage prep where they spray 6 phages on food prior to preparation and packaging to control listeria, and it's even considered kosher. It's even, and they also have one for E. coli 0157H7.
Do they? Oh, really?
Yeah. And
where do they put that?
They're going to do that in hamburger production. Because remember, a lot of the ground beef that's sold is made in slaughterhouses rather than butcher shops. And so they spray that into the grinding process so that your ground beef does come with phage, but they take out the E. coli 0157H7. And as the CDC points out, there's approximately 76 million illnesses each year in the United States from food and 5,000 deaths. And the good news about this salmonella outbreak with the peanut butter and the other nut butters is that only 30 people have been infected up to this point in time. The CDC has caught it, and only 4 persons have been hospitalized, and no deaths, fortunately, have been reported. But this again highlights how vigilant public health departments are to foodborne illnesses. And but think about the waste of how much peanut butter is going to be destroyed from this fact. It's a lot. It's been on the national news every night this week, and they've been interviewing the CDC. But here we have a natural product, phages. So this is not an antibiotic. I mean, this is natural. The rabbis have even considered it to be kosher in the case of listeria and in E. coli. And so I think, looking at the issues associated with producing food for an ever-increasing population on the planet, we really have to think about how Mother Nature has been controlling bacteria in specialized niches. And peanut butter could be just like a high-oiled specialized niche like we saw with P. acnes.
The whole control of foodborne outbreaks in the U.S. has really developed. I went to a session at ICAC in San Francisco on just foodborne outbreaks, and the CDC has an incredible system in place where they communicate with, local health departments at all levels. It's really, really very efficient, and so they can really pick up things very quickly, and they focus on certain kinds of agents, and they can move very rapidly. So I'm not surprised that in this case, they got on it right away. It's really very interesting.
And it's Salmonella Bredney, Bredney.
Wow, interesting. Okay, that will do it for 2 nice papers. We have just a couple of emails here I'd like to read. One is from a fellow named Jake, who writes, I've been listening to your TWiM podcast now for a few weeks, as I am a student at San Diego State University taking a microbial genetics class with Stan Malloy.
How about that?
Yeah.
The dean even teaches. I'm really impressed.
Yeah, he does research too.
It will be another few months until I've caught up with the current episodes. I saw this recent article on phage use in cleaning up acne and thought you'd like to hear about it. So there you go, Jake. This is an article in Genetic Engineering and Biotechnology News. And listen to the title, guys. You're going to love it. Zapping Zits with Viruses. So it's basically the story we have done today.
Jake, you're spot on.
Spot on. That's right.
Be more timely. Thank you.
This is a letter from Franco, who is in Italy, it looks like. And he writes, the article by Moule et al. In the episode The Sound of Whooping Cough is contradicted by a study published on the current issue of Emerging Infectious Diseases. I would greatly appreciate a comment on this point. So we had linked to an article on the CDC was it emerging infectious diseases? Let me see. Let me get this right. We had linked to a CDC article which said Bordetella pertussis strains with increased toxin production associated with pertussis resurgence. So that's the article by Moule et al. So they're saying basically that there is that some of the strains have a novel allele for the promoter on the toxin, and they get more pertussis toxin production. And then he linked to an article in Emerging Infectious Diseases, which said that basically there is great homogeneity in the B. pertussis population in the study that they've looked from 1935 to 2009, looking at isolates collected. So, I don't know, Michael, what do you think? But those are two different things, really, right?
Yes. The devil is in the detail. And I haven't had time. I saw the comment by Franco, and I went out, but I didn't quite digest the statistics between one and the other.
Yeah. Because I think if you're looking at population diversity with, probably multi-locus sequence analysis, right, Michael? That would be the
both of those the paper that he is citing in 2012 is both using MLST and then MVLT, where they're looking at the variations,
right.
the short, sort of like CRISPR sequences, except different.
Yeah.
So I'm going to take a look at that over the weekend and maybe talk about it on the next TWiM.
All right. That sounds like a deal. And then this next one has to do with an episode we did a while ago. This is from John. He writes, Dear TWimmers, I just finished listening to TWiM 39. Yes, I'm behind. And I had a question about the paper Michael reviewed. Let me spell out how I understood the discussion, and then maybe you can tell me if I've misunderstood something or if my question even makes sense. Michael was talking about the question of how mutations arise and are selected for in nature. The idea supported by Darwin, though long before anyone knew about DNA, was that mutations were in some sense driven by stress, right? And then there was this famous experiment which showed that mutants were present in the population before selection pressure was applied. A bunch of microbes were put on a plate with some antibiotic. A small fraction of them were mutants that had resistance genes. In this case, there was no pressure on the microbes to adapt to the antibiotic until they were put in a situation where they died unless they had the resistance genes. Is this right so far?
Well, the issue here is that the gene was there, and it does that they already were adapted. The only issue is you asked the question, do they have the gene?
Right.
And they answered in the affirmative, yes, we do.
Right. And presumably the genes were present maybe in nature to give the bacteria some advantage, right, under some conditions?
Yeah, absolutely. The genes were present in nature because antibiotics are natural products produced, in the case of penicillin, by fungi. And so consequently, microbes through time have probably been exposed to it, and so that what does not kill you makes you stronger, and there must be some selective pressure to carry that gene, albeit for the most time in this particular case silent, but it's still there.
Right,
and the beta lactamase, you just asked, do you have beta lactamase, and the answer was yes.
Okay. Then there was a second experiment which used less powerful selection. The microbes were put on a plate where the only carbon source was lactose, which the normal population of these microbes couldn't use for carbon. At first, they didn't grow much, but over time, even in an environment where the normal microbes couldn't grow at all, and so selection shouldn't have been able to bring anything new about, some microbes arose that could grow under these conditions. Again, have I understood the basic idea? What do you say?
Well, this is where it becomes inside baseball. It all goes back to promoters, and the Lac Operon is they begin to teach the Lac Operon now in grade school, but they leave out the hard parts. And the hard parts is promoters are leaky. And occasionally an RNA polymerase does indeed find the active promoter region for lactose, and you make a few molecules of beta-gal and you make a few molecules of permease, and you can continue to bring lactose in. So again, E. coli normally has this gene, and normally it's off, but I guess I'm a bit confused as to the question.
Well, he wants to know if you can get microbes growing on lactose, sole carbon source lactose, which is not a matter of selecting mutants because there's no growth initially. But I think, as you said, sometimes there's leaky promoters. Right?
Yeah, and that's the Roth argument.
Right.
Basically.
Now, this is where I get a little confused, he continues. From Michael's explanation, what I got was the idea that a bacterial cell can mutate in place even when it's not dividing. That is, even in these bacteria that were sitting there unable to divide because they couldn't get any carbon from their environment, mutations could arise over time, and this led to a population of microbes that started out without the ability to grow with no carbon sources other than lactose, and then a few mutations arose that allowed some growth. Is this right? Is this kind of mutation important in eukaryotic cells, say in cancer or cellular senescence?
Well, this is really John Ross. And Elio, do you want to comment?
Well, I all I know is that things happen in a stationary phase when cells are not dividing. They are a little bit mysterious. It's not possible to know if they're actually dividing to some extent, and some are dividing and some are dying. So the total number stays the same. It's really difficult to know, and I think what happens under these conditions is still murky. What is true, what you said about leaky operons means that the cells are not totally shut off. There's plenty of metabolism going on, but we just don't know we don't know enough about it, really, is the answer.
Okay.
Well, one of my old mentors, Arthur Koch, always drilled into our thinking that the bacterium is an individual. And what you're measuring in the growth phase is a population.
Yeah.
And every bacterium starts its life cycle just like we are born, so are bacteria. And they go through their lives interpreting their environment, and their environment is what they perceive. The population sees something very differently. And if you think about pH being able to control some aspects of the growth curve that we as microbiologists visualize by either looking at viable counts or looking at optical density, it's really a population that we are looking at as opposed to an individual. I think some of the new technology in microfluidics will allow us to get at what Elio is leading up to, of what is actually going on, and being able to detect some of these events, because it's really a combination of an individual and a population. And recall, the founding principle of microbiology is that you have a single cell that forms a colony on a plate. So one individual becomes literally many millions of individuals from that one cell. If you are buying the concept of dilution to extinction, and then the single cell becoming a colony.
Mm-hmm. Right.
By the way, Art Koch and I go back a long ways together, so I'm glad you mentioned his name.
Oh, Arthur was phenomenal to my training.
Person.
He was phenomenal in terms of cell division. He's one of these eloquent communicators like Elio in terms of how he first explained to new graduate students how penicillin would work and how you could explain it to your mother. And that is really a funny story. Arthur always carried a Swiss Army knife with him wherever he went, and he said the way to explain penicillin activity is as follows. You get yourself a balloon, and you get yourself a nylon stocking from your girlfriend, and you put the balloon into the nylon stocking and you begin to blow it up. And so you're blowing the balloon up in the nylon stocking. Then reaching into your pocket to pull out your penicillin, and he pulls out his Swiss Army knife. He then proceeds to cut the cross-stitching of the nylon stocking, which then causes the stocking to proceed to run. And he's doing this all live while he's blowing up the balloon. And he continues to blow up the balloon, and as he continues to blow up the balloon, the nylon run continues to expand, and he keeps blowing air into the balloon, illustrating to us that that penicillin requires actively growing cells, and eventually the balloon bursts because the nylon stocking has so many holes in it, it's no longer able to contain and support the increased air in the balloon. And that's when he points out that the average pressure inside a bacterial cell is the equivalent of a bicycle tire at 80 pounds per square inch. And I'll never forget that story because it was so memorably graphical, and yet it was so correct.
And of course, at the end, his girlfriend's pissed off that he ruined
one of her stockings.
And then you can't, and now you can't do that anymore because they don't make nylon stockings, right? And they wouldn't work with the synthetic stuff because they don't run.
They don't run. That's absolutely correct.
That's great. That's very good. You actually saw him doing this, Michael?
Yeah, he did this when we were good students. Yeah.
All right, let me finish up John's letter here. He says, an alternative seems to be that the bacteria started out with a huge diversity of mutations already present, many of which gave them some small ability to get carbon from the lactose. Then over time, it seems like those would do all the dividing, and you'd get something like classic selection with the bacteria that could reproduce faster, leaving more copies of themselves, which themselves could mutate to get better at reproducing. Was there something observable in the experiment that ruled this out?
I think so. I think it makes sense.
It could be. Right, Michael?
Yeah.
Anyway, thanks for answering my amateur questions.
It wasn't an amateur question.
I'm a computer scientist with no biology background, so I apologize if I'm making some dumb mistakes somewhere. No, that's pretty good. I think he's understanding it well. I also wanted to point out a really fun quote I ran across a while back. It's in a very old book called Mutual Aid, A Factor in Evolution by Peter Kropotkin. I think it was published in 1902. It was a
Russian prince who was an anarchist.
Is that right?
Yeah.
It was a kind of argument against social Darwinism from a Russian naturalist and anarchist, combining observations on history and nature both. And the quote is, mutual aid is met with even amidst the lowest animals, and we must be prepared to learn someday from the students of microscopical pond life facts of unconscious mutual support, even from the life of microorganisms.
Wow.
Amen. Amen.
Reading this, the words biofilms and quorum sensing popped immediately into my head, along with all the other kinds of mutualism between microbes. Thanks again for your wonderful podcast. Nice letter.
Thank you, John.
Much. Thank you very much, John. And I think we will stop there and finish with the others next time. You can find TWiM at iTunes, the Zune Marketplace, and microbeworld.org/TWiM. Do send us your questions and comments. You can email them to twim@twiv.tv. So that's TWIM@TWIV.tv. Thank you so much, everyone, for participating today. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure. Fun as always.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
You're welcome, Vincent. It's been fun again. And Elio, have fun with Stanley.
Oh, I will. No doubt. Thank you.
Michael, do you carry a Swiss Army knife?
Not since I fly so much. I used to.
Yeah, I was going to say the fellow you mentioned would not be able to carry it with him all the time nowadays. Take it away on planes. I'm Vincent Racaniello, and you can find me at my website, virology.ws. Many thanks to the American Society for Microbiology for supporting TWiM, Barbara Hyde, Chris Kundayan, and Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.